just got finished talking about the domain Eukaryota. But there's two other domains which include the prokaryotes. So there's domain bacteria, also called eubacteria, and domain archaea. And these are the prokaryotes. So prokaryotes is not a domain. It's what the two remaining domains are included in. We talked about prokaryotic cell compared to eukaryotic cell in the last unit. So let's just read through this a little bit. Bacteria are everywhere. They live everywhere. In the air, in the water, on surfaces, everywhere. In our body. Now, some bacteria are pathogenic. They cause disease. But that's not all of them. Most bacteria are non-pathogenic and even useful. We have bacteria living in our large intestine that helps us to absorb vitamins from our food. The archaea live in hostile environments, often found in extreme environments, places where there's no oxygen or no light. Real high pressure places like deep in the ocean, or really low pressure places like high in the atmosphere. Places where there's extremely cold or extremely hot temperatures, like hot springs or glaciers, ice cap ice, places that are very acidic or very basic, places that we could not survive in for sure. So archaea is the second domain of prokaryotes that live in extreme places. Some archaea are found in our gut because that's a place where there's no oxygen. I said gut, large intestine. So once again, most bacteria do not affect us. They're not pathogens. Some help us, like I said. The ones that live in our large intestine help us digest our food and absorb vitamins from our food. So this is stating that we have about the same number of human cells in our body that we have bacterial cells living in it or on it. Bacterial cells live in a symbiotic relationship with us. It's a beneficial relationship for us because those bacteria produce products that help us digest our food, but also that keeps the pathogenic bacteria away. Bacteria produces antibiotics so that they can kill other bacteria that are infringing on their territory and competing with them. So bacteria are also used to make yogurt and cheeses. They give that flavor to the yogurt and cheeses. different cheeses have different flavors because of different bacteria being added to the milk that the cheese or yogurt is made of. The archaea, again, is a separate domain from the eubacteria. Most of them are extremophiles. They live in environments where other forms of life cannot live. Like I mentioned before, extremely high or low pHs, extremely high pressures or low pressures, extremely high or low temperatures. Very salty or very places where there's a lot of toxic metals like mining outflows. Scientists think that the first cell that existed was obviously a simple cell, like a prokaryote. But who came first? Who evolved first? Was it archaea or bacteria? Today, we think it's archaea for several lines of evidence, which are kind of technical. So we believe that archaea was probably closest to the common ancestor of all life, the first cell. So we are more closely related to archaea than we are to bacteria when we compare the genes in the DNA, which we'll talk about later. Now, like these right here, this picture shows deep in the ocean where there's cracks in the crust. Underneath the crust is this hot lava. Melted rocks is so hot, called magma. And, that stuff oozes to the crust underneath the ocean. And when it enters the ocean through a crack like this, it boils that water, and a whole bunch of minerals that were in the lava get spewed out and giving it looks like a plume of smoke coming out, but it's really hot water filled with minerals. Okay, now, very hot. Now, down here it's really cold, the ocean, because no sunlight gets to it. But right here, these really hot spots, these are called hydrothermal vents. Or black smokers. Slang. Okay, but we get all kinds of minerals coming out in gases. And they fall out, and they provide food and nutrients for bacteria that grow, that can get their energy from these minerals. And then there's eukaryotic organisms, multicellular organisms that can eat that bacteria. And there's a whole food chain down there in this dark, super high pressure place at the bottom of the ocean. So we think life originated at these hydrothermal vents. Obviously, life did originate in the oceans because there was no dry land. when the Earth was forming. It was a ball of very hot crust covered with water. And here's a picture, an actual picture of a black smoker or a hydrothermal vent. And you can see all the minerals pluming out of the ground there. And right here is the rock where the minerals solidify. Look like little miniature mountains. So here's a phylogenetic tree. And it looks like here's where we are, eukaryotes. All the red things are eukaryotes. And we can see that from the common ancestor to all life, the archaea and the eukaryotes are more related than the bacteria, which are the blue ones, the eubacteria, and the eukaryotes. So, because this branch on the phylogenetic tree, evolutionary tree, is closer to where we branched off. We're more related to archaea than we are to bacteria. Now we know this because the archaea genes and the way they make proteins from their genes is more similar to how eukaryotes do it than how bacteria does it. Okay, so the earliest living organisms were most like archaea. All bacteria are harmful. Absolutely false. Prokaryotes can live in which of the following environments? Anywhere. Anywhere. So all the above and more. This question is kind of difficult because bacteria live everywhere. But think of a place that is warm, has lots of nutrients. Desert sand is warm, but not very many nutrients and very hot. So probably not. Jar sugar is one substance, glucose, and if it's in sugar, it's not dissolved in water, so it's not really available too much for bacteria to eat. So I would say no on that one. How about a cup of swamp water? Like pond water out there in back of our classroom? Yeah, that has probably a lot of nutrients and a lot of bacteria, so I would pick that. A closed packed dried fruit. Well, dried fruit, again, the sugar is not dissolved. It's probably, there's bacteria on it, but it's not going to be, it's not as available. The nutrients aren't as available in dried fruit as in a cup of swamp water. There's preservatives in that dried fruit too. Okay, so our digestive tract, our gut, specifically our large intestine. What's the purpose? How do we rely on bacteria? Remember, there's a symbiotic relationship. We provide them a nice warm place with food, and what do they do for us? They help us break down our food. Yeah. They produce gas in the gut, causing an upset stomach. Well, some do, but it's really not. If it's in your gut, okay, but that's not why we rely on them. Some pathogenic bacteria in food that cause food poisoning can do that, I guess. They create antibodies to destroy other harmful bacteria. Well, that's definitely true. So A and C are correct. Okay, bacteria and archaea are unicellular. There's no multicellular forms of bacteria. When you put bacteria in a place, or they find a place where there's food, that one bacteria will divide into many, many bacteria and produce what is a colony of individual cells that are exactly alike, clones of each other. So here we have a petri dish. And the petri dish has a growth medium on it, which is just a gel called agar with nutrients dissolved in it. It's like a jello. And it's like a stiff jello. And then the bacteria are growing in colonies here. There's millions of bacteria cells in one colony. They'll cover that whole plate if you let it. Okay, there's different shapes of bacteria. You should know some of these. Kasai is spherical. Bacilli is like rod shape. Then we have some other weird shapes, comma shape, corkscrew shape, club shape, helical. Okay, anyway, so those three you should recognize: bacillus, corkscrew, and kasai. Okay, we already talked about this. We were comparing eukaryotic cells to prokaryotic cells, but let's add some more stuff, which I avoided. Okay, so the nucleoid region, there's no nucleus, right? But the nucleoid region is where the DNA is. Yeah, there's one DNA is that blue squiggly stuff. So I'll call that that's the DNA is a single circular chromosome. It's in a singular circular chromosome. Doesn't look like a circle because it's all tangled up. There are ribosomes. That's the only organelle that prokaryotes have. It has, it can have, well, all of them have cytoplasm. Cytoplasm is really cytosol, everything in it, like the DNA and the ribosomes. Let's see, it might have a whip-like appendage called a flagella. But really it's called flagellum for singular. But that allows us to propel around, to move through watery solutions, like a little propeller. Okay, pili. Pili are basically for fixing to a surface, adhering to a surface. like your teeth, so they can dissolve your enamel. And what else? We got cell membrane, of course. A cell membrane, I don't know why they call it cytoplasmic membrane or plasma membrane. You can see that right there, that gray envelope around the whole cell. And they do have a cell wall, but it's not made of cellulose. It's made of a different substance, not chitin either. Let me just write that. Cell wall is made of peptidoglycan. Peptidoglycan, which is a carbohydrate. Cell wall is made of that substance. And then it has a capsule, like a slimy coat. It has a capsule as well, a slimy coat over the cell wall. So here we have what I just talked about. I talked about the capsule. Slimy, I call it, but it's sticky. Helps it to stick to whatever. With the pili, it helps it to stick to a surface. And then the flagella, which allows it to be motile. Motile means it can move on its own power. And it's powered by the flagella. So, why it's important to be able to move so that it can move towards light if they're photosynthetic bacteria, or move towards chemicals that they can get energy from. The ability to move towards or away from bad things, towards good things or away from bad things, is called a taxis, like a taxi. So chemotaxis is moving towards nutrients, and phototaxis is moving towards light, where it can do photosynthesis. So we talked about pili. Pili, I didn't really say they're protein tubes. They're extending out of the cell through the cell membrane, of course through the cell wall, and then through the capsule as well. There's two kinds of pili. The ones that are really numerous are called fimbriae. They allow it to attach to a substrate, to something like teeth. And then there's pili that are longer, and they're called the sex pili, or the F meaning fertility pili. You have a whole bunch of fimbriae for fixing yourself to a surface, but if you have any, you only have one or two sex pili, and these are longer, and they allow the bacteria to transfer genes through the hollow tube from one cell to another. And we'll talk about that a little bit. So it looks like this guy right here is donating some genes to this recipient over here. I don't know why this guy doesn't have fimbriae. He should. So, some bacteria don't have the things I just mentioned, even a cell wall, depending on what type of bacteria they are. It depends on what environment they live in, whether they have those adaptations. But of course, all of them have a cell membrane, all of them have cytoplasm, all of them have DNA, a nucleoid region, all of them have ribosomes. Okay, so the things I checked, every prokaryotic cell has. Okay, this is a repeat of what I've already said. The chromosomes are free floating though. You don't have any rough ER, any of that. You don't have any membrane bound organelles at all. The only organelle you have is ribosomes, and they don't have membranes around them. Okay, you can read this, but I already mentioned this many times. Okay, what structure allows a prokaryote to stick to an environmental surface? Those are the pili, but not the sex pili, the fimbriae. Okay, taxis is moving, being able to move yourself towards something favorable or away from something not favorable. That would be the flagella. Flagellum is singular for flagella. Okay, peptidoglycan is the substance that makes up a cell wall of a bacteria. With the shape of a bacterial chromosome is a big circular loop, but it's all tangled up. Okay, prokaryotes have only one chromosome. The region where the bacteria chromosome is located is called the nucleoid region, or just nucleoid. Okay, a little review here. Bacteria have DNA just like eukaryotes have DNA. Same kind of structure. The genes are basically the protein recipes. The information for making proteins is coded in the order of the nucleotides. A DNA molecule in bacteria has every recipe for every protein that the bacteria needs to make. So, one gene is a protein recipe. There's only one sex of bacteria. There's no female or male. There's no sex to a bacteria. If they want to reproduce, they're going to have to do asexual, without. A means without. Asexual reproduction. So one cell decides to just hum-de-dum, split up into two, forming two daughter cells. Okay, these daughter cells are genetically identical to the parent cell. Here's the parent cell. We call these daughter cells. Okay, first the DNA has to double itself in the parent cell before this can happen. And because the children, the daughter cells, are genetically identical to the parent cell, they're called clones of each other and of the parent cell. We call this asexual reproduction that bacteria undergoes. We call that binary fission. Binary means two, fission means splitting. Now, before the cell splits, it has to replicate, or copy its DNA so that each daughter cell gets exactly the same amount of DNA and same kind of DNA. So the next slide is going to show you what this slide is talking about. The chromosome will start self-replicating, copying itself. Replication bubble, site of origin. That's not really important, but it's interesting. Let's see the next picture. Okay, so the origin is where DNA starts replicating before cell division, and then each side it opens up, and then the dark purple is the original strand, and the light, called the parental strand, and the light colored one, the light colored purple, is a new strand that copies exactly from the old strand. It's called the daughter strand. And so daughter strand, this keeps on going until you get to the end, you're done, termination, and you get two rings of two chromosomes, each one with half the parent and half of the daughter, half the new DNA connected. Okay, so that's how the DNA replicates. And I should mention that right here is the replication bubble, and you can see the replication bubble is getting bigger and bigger until it's the whole size of the ring. So replication bubble is what I'm showing in gray. I mean, for what that's worth. It expands as the DNA replication continues. Okay, so here's a straightened out picture of what's happening. You can see these little Y at the end of the replication bubble, or replication forks. Okay, we call this semi-conservative replication. That's what's important, where each new chromosome that forms is half the original and half the new DNA. The parental strand and the daughter strand. This is called semi-conservative replication. And we do the same thing in our eukaryotic cells. Semi-conservative replication. Okay, after the chromosome is replicated, the cell will then divide, pinch in the middle, and divide into two new cells. What type of organic compound is DNA? This is nucleic acid. The twisted ladder or spiral staircase structure of DNA is referred to as a double helical shape. DNA is made of chains of nucleotides. The monomer of nucleic acids are nucleotides. The location of a bacterial chromosome where replication begins is known as the starting point. I would say starting point, but it's called the origin of replication. Okay. At the end of binary fission, that's when a bacterial cell, divides into two. There are two prokaryotic cells. One has all the parent DNA. No, this is semi-conservative replication of the DNA. semi-conservative replication. So each daughter cell is half the parent. Each daughter cell's DNA is half parental and half new. So it's letter D. Half a parental strand and half a daughter strand. Okay, plasmids are tiny accessory ringlets of DNA that aren't associated with the bacterial DNA. They're not connected to bacterial DNA. These plasmids can have a purpose of resistance against other bacteria or archaic antibiotics, which are drugs that kill other types of bacteria produced by other types of bacteria. It can be that plasmid can give it the ability to form sex pillars. It can give it the ability to protect itself by making proteins that are toxic. Also, it can give them genes to protect them from poisons like heavy metals in the environment. But they're accessory DNA circles called plasmids. Okay, bacteria with our plasmids have antibiotic resistance. So if a population of bacteria is subjected to some antibiotic, the ones without the R plasmids will die. The ones with R plasmids will survive. Now, this is why it's important to if your doctor tells you you need to take an antibiotic and prescribes an antibiotic, and if you don't completely finish the prescription, let's say you feel better after the second day, and you say, I don't want to take that yucky tasting antibiotic anymore. Well, then what's happening? You didn't wipe out enough of them you don't wipe out enough of the bacteria, and they will come back because some of the antibiotic resistant ones or slightly antibiotic resistant ones will survive, and they'll pass around the R plasmid through their F pili, sex pili, and pretty soon you're sick again with a bunch of resistant bacteria that won't be affected by the same prescription of antibiotics. So the doctor's going to have to give you a different antibiotic. Of course, our plasmids lend resistance to antibiotics, so that would be a problem if an animal gets bacterial disease and the antibiotic won't work because of the resistance of the bacteria to that antibiotic. Okay, ability of prokaryote to act as a donor during conjugation is usually due to conjugation is when you connect with a F pili or sex pili. So you need the F factor, you need the plasmid, F plasmid, in order to grow sexpilus, sexpili. Okay, we're going to talk about viruses. Viruses are not bacteria. They can be germs too. They can be pathogenic, cause diseases, but they don't have to. But when we think of a virus, we think of something that causes a disease. But it's not anything like a bacteria. It's important to understand that viruses are not living. They're particles with some genetic code in them, but they're not living. They infect living organisms, and then they seem to get the characteristics of a living thing, but it's not them. It's the host cell that they infect that allows them to pretend that they're living. For 3 reasons, they're not living. They are not comprised of cells. They don't have cell membranes. They don't have cytoplasm. They don't have ribosomes. They have a little bit of genetic material, and that genetic material might be DNA or RNA. They're protein capsules with some genetic material inside. They cannot reproduce on their own. They can't. They need to infect the host cell, much like pirates invade a ship and take over it in order to do what they want. And so viruses have to infect a host cell in order to be able to reproduce. But they can't reproduce on their own. They don't have enough chemical equipment to do that. Also, they cannot break down food. They don't eat. They use the energy of the host cell. So they don't do homeostasis. They don't do all the things that a living thing can do. Okay, so they're much smaller than a bacteria, so each of these individual pirates invading the ship, the big thing is the bacteria, and he's the host, the unwilling host. And these little guys are the viral particles, or the viruses. All there is attacking a bacteria. Bacterial phages. Phage means eating. Each of those guys, and they're bacterial phages. So I'm going to show you a picture in a minute. The bacterial phage is a protein coat with a tail piece that connects to a bacteria. And there's a DNA or RNA and injects its DNA into the bacterial host. And that viral DNA genes will be able to replicate and form new viruses inside the bacteria using the chemical equipment of the bacteria that is invading. That those new viral genes that have replicated will form protein coats using the chemicals from the bacteria, and they will burst through the bacteria cell. All these new baby viruses will break through the bacterial cell and attack other bacteria. So here we see an actual picture, electron microscope picture of a bacteriophage, and inside this place is where the DNA, inside that protein capsule, is the DNA or RNA of the virus. Now, this is called the capsid. Actually, the protein capsule is called the capsid, and the DNA would be right there inside that hollow shell called the capsid. Okay, so a virus is very specific to its host. Viruses don't just attack bacteria, they can attack humans, the cold virus, the measles virus, the COVID virus, the mumps virus, shingles virus, HIV. Okay, there's two possible life cycles for viruses, and they're not really life cycles because a virus, again, is not living. There's two ways, two strategies a virus can take when it attacks a host cell. Okay, so first, let's start with number one here. He injects his DNA. The DNA replicates, or it could be RNA, replicates using the machinery of the host cell. The green thing is the host cell. It could be our cell, it could be a bacteria. Okay, then that all those new RNA or DNA particles, molecules will translate in their protein, the viral protein, using the host cell's chemicals. Then they'll assemble together. The shell will come around the new viral DNA or RNA and will all assemble together. And then they'll burst through the cell. It doesn't have to be a bacteria cell, it could be our cells. You've all experienced a cold where your throat gets those cells die because the viruses are coming out and killing those cells. Then the white blood cells clean up that mess and try to kill the cells with viruses. And that's when you get all that green snot. That's dead white blood cells and cells that have been killed because of the viruses. This is called the lytic cycle. The lytic means break, cut. And so that's what the viruses are doing. They're breaking through the cell that they infected after the job is done. The job being to make more viruses. So it's important that you realize that this will go, the lysogenic cycle will go into the lytic cycle, where that guy, the viral gene, will continue replicating itself and making new viruses and kill that cell. The lysogenic cycle, again, remains, like we said, the viral gene remains in the host gene, and the cell just keeps on dividing. Each time it divides, the viral gene is replicated as well with the host DNA. So eventually the lysogenic cycle will switch to the lytic cycle. Transduction is when the lytic cycle occurs, and what happens is, let me show it. Okay, so there's a cell. Here's the bacterial gene, or R gene, and here's the viral gene. Okay, so transduction occurs when, well, before we talk about transduction, this will produce, this is the lytic cycle, where when that gene is, when that DNA is replicated, so is the viral gene replicated. So eventually, and that will keep on going on and on and on. No problem, you don't feel sick at all. Bacterial cell is unaffected, or our cell is unaffected. But eventually, the lytic cycle will kick in after that little green virus pops out. So what happens is the green virus will pop out, and pretty soon, once it pops out, it makes a whole bunch of them, and then the cell breaks open after the protein coats are produced and then assembled. The viruses are assembled, so you get this cell breaking up and releasing all these little viral particles. Okay, this step right here is the lytic cycle. This step right here is the lysogenic cycle. Now, in the lytic cycle, when these green things come off, when the virus comes off, it takes some of the host DNA with it. I'll draw like a little black bit. Then what's going to happen is you're going to have some of the host DNA in there. So what happens is if that viral particle with the host DNA infects another cell, host cell, which it will, and infects another host cell. Let's say the host cell, let's make that red, that DNA red. So what's going to happen is when he infects it, the lysogenic cycle happens, and you're going to get the green virus incorporating into there, but you also get some new genes from the host genes. This is called transduction from the host before. So this is the previous host cell, the previous host genes, maybe one gene or two genes gets ripped off. This is the viral gene, and there's the new host gene. Genes. Okay, so this idea of getting the previous host genes incorporating into your genes, the new host genes, is called transduction. And that might be a benefit because that adds genetic variety to the host. It could be resistance to bacteria. It could be some gene that is going to be a gene that produces an adaptive protein that increases the survivability of the host. So transduction could be a good thing for the host cell. Okay, viruses that infect bacteria are called bacteriophages. They eat bacteria. Phage means eat. When a virus infects E. coli, that's a type of bacteria cell that lives in our gut. What part of the virus enters the bacterial cytoplasm? Just the DNA or RNA. In the lytic cycle of phages of viruses, the cell typically dies, releasing many copies of the virus. That is true. The entire phage, no, just not the protein coat, enters the bacteria. DNA replication is part of the life cycle. Well, yeah, it is. DNA replication of the virus is part of the life cycle. Phage DNA is incorporated into host cells' chromosomes. No, that would be the lysogenic cycle. So, yeah, DNA replication is part of life cycle. So, both these are true, and there's kind of a mistake there. DNA replication of the viral gene. Maybe they meant DNA replication of the host gene is part of the life cycle, then that would be wrong. But if you think of DNA replication of the viral genes as part of the life cycle, that would be very important. The host cell dies in the lytic cycle. Eventually, the lysogenic cycle may go to lytic cycle, but that's where the host cell gets burst with all the new virus invaders leaving. Which cycle can be utilized by temperate phases, but not by many other. Okay, temperate means it hangs out in the host DNA. It's like what do you call that? A spy, that undercover agent. He hangs out. So that would be the lysogenic cycle. Which cycle results in production of full virus molecules? Well, that's the lytic cycle. The lysogenic cycle, if it goes to the lytic cycle, will also produce full virus molecules. But it's really the lytic cycle part that happens. Which cycle makes it possible for transduction to occur? Okay, that would be the lysogenic cycle. Okay, end result transduction could be that one if you don't go through the like like the lytic cycle. If you don't go through a lytic cycle, the new host cell will take some, very very little, but some. So that's true. And then this one could be happen if eventually the lytic cycle occurs. So I would say B and D. They probably mean B because you don't necessarily have to go lytic.